Very nice to meet you all. For those who don't know me, I'm Giulia. I'm currently finishing my first postdoc here at UCL that I joined after finishing my PhD in Oxford with Ellen. I have to say, there's a spoiler, I'm not really a, how do you say, cell movement person. So I just recently started working on this. This is something that I started at UCL together with Jonathan Chapp's lab. And yeah, I'll tell more about my main interest later on. But so there is a lot that I've been learning, and for sure you can give me lots of ideas on this. Before starting, I just want to thank you, Ford, who's the person who did all of the amazing experiments, which is, I think, one of the nicest bit about this work. I have to say, I was amazed by when they showed me their images. And this is work on understanding how Dictyostelium cells, which are common at amoeba, self-organize as they collectively chemotype. So for a moment, I would like you to forget what the title is and just look at this video and, just like think, what do you think it could be if you weren't to know the title? Okay, we really the segmented, but it's more fluent when you see it from my computer. But I have to say that if I had no clue of what was happening, the first thing I would think of is droplets of water. It seems like a fluid. What is actually going on is that I'll give you a little bit of an explanation. Now, from this type of images, it's difficult to see, but basically on the right of the trail, we have a row of bacteria cells that are the food Dictyostelium cells are feeding upon. And then on the left-hand side, instead, we have this moving front of Dictyostelium cells that as a swarm collectively move, but as they do so, they leave behind quite periodically and very nicely this group of cells. And so
the food is where we can see sort of white coloring.
It's like you can't really tell the difference now, but I'll show you afterward because here is Saharan images. So actually, Dictyostelium cells and bacteria kind of seem the same, but yeah, it's basically where there is the vertical line is basically more or less where the front of this form is. What is very nice is that they can actually do lots of imaging on this system. This is a little bit of a different setting. But so on top you see now we don't have just a trail of bacteria, but we have an entire lawn of bacteria cells that you can see highlighted in light blue. And then instead, the cell nuclei of Dictyostelium cells are highlighted in orange. And you can see that even in this setting, there is this nice periodic pattern that emerged. And in this system, there is no curing mechanism for patterning. So it's a new way in which the cells can kind of self-organize as they move collectively as they feed, but also create some leaving behind some patterns of cells. And then they can also on the bottom, you can see if you were to take a cross section of the first image and look from the side, what you can actually see is the flow field of the cells. So here they're able to follow individual cells. And again, if you were to look at these without knowing what I'm talking about, probably the first thing you would think of is Poisson-Le flow, which is naturally emerging in fluids, in viscous fluids. So they have an amazing set of data, and the idea is that looking at all of these, you would think like the first assumption you would come up with is that, oh, Dictyostelium cells are behaving collectively as a fluid. But one of the major open questions that we have is like, why they do so? How does this emergent property of viscous-like fluid comes in? And then also, what are, so this is our open question. Well, we have focused more on understanding what are the mechanisms that give rise to this periodic shedding. So what are the forces? What are the mechanisms at play? Can we explain what's happening from a continuum level point of view? Because these patterns are like macroscopic patterns compared to the size of individual cells. But yeah, there is the open question of like, how does all of these emerge from individual cell? So I'll tell you what we have concluded about question 1, and then the key idea of the open question is really question 2. So what are the key mechanisms at play? So what we think is happening, what we think are the key mechanisms is first, the Dictyostelium cells are proliferating. So this is like normal fluids, is a fluid that is increasing in mass over time. And then what's happening is that at the individual level, if you were to consider individual Dictyostelium cells, what they're feeling is actually a chemoattractant that is emitted by the bacterium. So they have these volatile diffusing molecules that are produced by the bacterium, and they're sent by the Dictyostelium cells that, with this kind of external protein bonding. And then at the same time, Dictyostelium cells are also eating bacteria. So they're kind of self-generating gradients in the chemoattractant and in the bacterium concentration that then drives their movement. So they're moving up self-generated gradients. And then the idea is that unlike bacterium, Dictyostelium cells can't actually swim. So the way in which they move is that they form adhesion, like short-term adhesion between each other, and they actively pull on each other to move themselves forward. Yes.
Giulia, are you okay with interruptions?
Sure, I love that.
So usually people study Dictyostelium in starvation, right? And then
Yes.
it's active AMP as kind of a system.
Yeah.
So that's a different case.
That's true. So the idea is that CMP is key to the initial coming together of the cells, but it's not playing a role in this case. I can show it. It's not playing a role when they chemotact. So there is no there is no chemical signaling related to CMP that is responsible for the shedding. CMP is activated initially as they come together, because they are conservation, of course, to stand the bacteria. But indeed, what's interesting in this case is that CMP is not actually playing a role. And yeah, for people that work on Dictyostelium, that's the key idea behind collective organization. Well, here it seems like all cells behave the same. There is no kind of macroscopic signaling from that point of view. It's more like external chemoattractant gradient. So yeah, as I was saying, in terms of how they move, is that these cells are creating an active force. They're actively pushing on each other. And what we think is going on is that when there is a strong signal, so a strong gradient, cells are aligned. They're all trying to move in the same direction. And so if you were to think about it as cells pulling on each other as a dipole, all these dipoles are aligned because they're all trying to move in the same direction. And that can be described by having an order parameter within the pneumatic liquid community. It's called like pneumatic order parameter. Yes.
What you said. Okay. But are they interacting, or they should ignore each other and then just follow the gradient? No. So you see.
But they're sorry. If you look at this image, this is like you have like probably 20, 50 cells like moving on top of each other, and there is no way these cells can move without creating adhesion and pushing. That's the way in which they move. So basically, if you look at the videos, they are kind of it's like if you think about a swarm of I don't know, I can't think about anything else, but it's like they're all trying to move in the same direction by pulling on each other continuously. So they are interacting because I create an adhesion, sorry, and then the acting is the composite, so they create a pulling force, like bringing back the one in front of them. I don't know if you meant. I can try.
No, I understand.
Climbing over the top of each other.
Yeah, and pushing each other backwards.
So once you have a whole load of people
On top.
and the bottom ones are a bit stuck down, and then the top ones climb, and the ones at the very top can sort of
Move to, yeah, but
right over.
exactly by you basically
Yeah,
create
because
a
they've got
vision
all
of the
the
one in
things
front of them. So it's very chaotic. Like this is a average over a few time frame. So it's, but when you look at them, it seems like really they are kind of just like pushing on each other and moving forward. So there is some actual physical forces they're exerting. I'm not saying we fully understand what's
So-
going on.
So just
but-
to understand, so now if you go back, so this you show that. So in other words, the one that at some point are at the top, they're probably the one fastest, then they fall down and they will be
Exactly. So
over by the one coming behind.
exactly. So this is in the reference frame of the lab. But then if you look at the reference frame of the droplet, what you would see is
Something
basically
like that.
a rolling. Yeah, exactly.
Right?
They have to interact with the substrate, otherwise you have a group of people hanging on top of each other and they fall.
Yeah, exactly. This symmetry is broken by the, so the,
As that round moves, does it leave some of the ones that are at the bottom of the pile behind?
yeah, in this case, this is a frame in which there is no shedding. So here is the flow field in the absence of shedding. When there is the shedding, of course, it's a little more complex because you can have some going back behind or backwards. But yeah, in some sense, like as they move at a certain point, there is some instability or something, they can't keep together, the droplet cannot stick together, and some of them like are left behind. But yeah, going back to your question exactly. So the idea is that if there was no surface, since they can't actively move, actively swim, these cells will not move. It's the presence of the surface that breaks the symmetry that allows them to move. So there is an interaction with the surface also in the model. That's correct.
Or are they just on
They're in a petri dish, soft gel.
with air or?
I think so. Here, yes, but for the imaging, there is some oil because the oil is necessary for the contrast for the image. But since we observe that also in air, we don't think it's the medium is so important.
They also move by slime. Slime trail.
So I'm not sure these are yet in this line mold kind of state. What they see that afterwards eventually they form protein bodies. So there is some kind of phenotypic transition in the system. Behind this droplet left behind. But that happens on a longer time scale. And that's the question of why would they do that? There's some idea that maybe it's related to that. Perfect. So yeah, exactly. There is some interaction with the surface, but the key idea is that we think in some way the amount of alignment of the fluid is dependent on the amount of information that they have about where the bacterium are, which is given by the chemoattractant. So if there are strong gradients in the chemoattractant, cells have a clear information about where they want to go. Where instead there is small enough gradients, these cells kind of move randomly. So some want to go right, left, and so on average don't move, and there is no enough order. And so the key idea is now that this cell-cell liaison that they form and these active cooling forces is modeled as typical active fluid with an active dipole. Cells. And so when we do so, these are, I'm skipping over a lot of things, but I can give you the details if you want. So these are some simulation of our model. On the left, what you see is like the simulated droplet front moving in the reference frame of the front. So they're actually moving in this direction, but you would see the front same things. And on the right side, you would see the mass of the droplet. The color blue is, oh, sorry, is the alignment. Is the alignment field. So basically, as I was saying, how much cells want to move in the same direction. As you see, initially they all move compactly as the mass increases, but eventually as we go above a certain critical mass, they elongate, and then eventually the elongation leads to the breaking up of the front and forming like this nice droplet on the back. And so the key idea now is that there's some sort of mass. If the mass of the droplet is below this critical mass, the cells can move as a compact droplet. And since growth happens on a much slower time scale, what we actually find is that the droplet is moving as a traveling wave. with a slow increase in mass that changes the velocity. But beyond this critical mass, the droplet cannot keep its compact form anymore as it starts elongating. And as it starts elongate, eventually, because of surface tension and other forces related to the surface, it breaks apart. And then it goes back into the cycle. And so, what is this critical mass? Can we kind of have an angle of it? What we do is that we actually study traveling wave solution of the system. And we study traveling wave solution as a function of the swarm mass. So you're going to have the velocity, the length of the swarm, and the shape of the droplet depends on the swarm mass. And what we find is that above the critical mass that matches the simulation, we have a bifurcation. We think it's a settlement bifurcation, but we haven't studied really properties because we are in higher dimension and we do that numerically at the moment. So we just know there is a bifurcation. And so what we think is happening is that initially the mass increases slowly. And so what the droplet does is that it moves up this green curve. So it's kind of a quasi-equilibrium, let's say, but in terms of a traveling wave system. But then eventually it gets to our bifurcation, bifurcation. And what it happens is that it needs to move out of equilibrium. Something needs to happen. And it jumps. And instead of jumping over to the other by traveling wave solution, so the other branch, because of the dynamics and the elongation, is instead breaking up, and then it goes back into the cycle.
Are you going to show the equation?
I can show you the equation.
Yeah.
Sorry, but when you say start again, but actually, I mean, does it move horizontally? So
we can't really tell where it goes back to. We just know that it goes back depending on the mass that it leaves behind. And so the mass that is left in the front is going to tell us where we go back to. But we can't really determine that. What we can just explain is these two modes of migrations. The first mode, in which the droplet is compact, can be well described by traveling wave solution. And then the reason why it breaks up, we think, is because there is this transition to the elongation, it's because of the presence of this bifurcation. Then the reason why it breaks up needs a little bit more of analysis, because it cannot be found. The breaking up is a very dynamic mechanism, so it can't be captured by traveling wave analysis. Because if you think about it, the front is moving at the velocity. When it elongates, the front is moving at a different velocity as the back. So there is not actually a velocity traveling wave reference frame that we can follow it on. And so one of the things we want to do actually now is use some asymptotic analysis and try to actually analytically describe this bifurcation. But that's yet to be done.
The bit that's shed, does that just sit there and it doesn't proliferate or anything?
Yeah, no, because there is no bacterium anymore.
Right.
And if you look also at the footage, on a short time scale, these things remain and they look like a passive droplet.
Yeah.
In the longer time scale of the experiments, eventually, either the form fluid body of the move parts, but there is some kind of phenotypic change. But when you look at them at the short time scale after the shedding, you just sit there and they look like lovely passive droplets for some reason.
Like this is actually a starvation medium with some food in the front.
Yeah, exactly. Yeah, exactly. And that's why they come together initially. And that there needs to be, in order for this droplet to form, they need to be above a critical density. Otherwise, they would move independently. But yeah, exactly. Like initially, there is no food, they come together and then they start moving up this lower bacterium. So, okay, I'll tell you this afterwards. So if you, a little bit about the modeling, and then I'll go and show you the precise equation. But so the idea is that we model this droplet as a thin field. So it's a very thin droplet. So the length is much longer than the height. And so we can use lubrication theory model it. And the key ingredients are the ion. We describe the droplet by its height, H. We look at the 2D cross section. We have some surface tension. We then have the flow field that describe how cells move inside the droplet, that's we see. And then there is this extra term, which is the active stress that is typical in the pneumatic active fluids that comes from the idea that cells are fully actively pulling on each other. So if you think about water, in order for water to move, you need to have an external field, an external gravity field, for example. What we can happen here is that energy is generated at a macro scale because the cells are interacting with each other, and by interacting, generate these active stresses that can drive the flow. So if you look at the equation on the bottom, this describes how the cell velocity defines the cell velocity, but then dictates how H changes over time. And we have to confirm the first one is dependent on the surface tension, and this is typical of lubrication theory, and is a capillary flow. But on its own, the capillary flow cannot drive the system anymore. What is driving the system more is this actor in the physics community, and this depends on an activity side, which is in this case negative. The cells are pulling instead of pushing on each other, and then depends on the gradient in the polar in this aligned order parameter S. And if you recall, what I said at the beginning, S depends on the gradient of C. So basically, chemotactic comes into play by creating gradients into the active force, and this generates an effective flow of cells.
So what sort of functional form would F have? Is it?
So it goes to 0 at the bottom, and then it kind of saturates to 1. So the idea is that even these cells are very sensitive. So if there is a little bit of information, the student cells will just go for it. But that is when they when there is no information, so the gradients are too low, then S goes to 0. And so this eventually creates this gradient, this active force that leads to the cell movement. And then the other part is the interaction with the surface, and this comes into play by sleep condition. So cells can actually sleep on the surface, which allows them to actually move, and also the contact angles. That's really tricky. And then if you look at the flow field in the reference frame of the droplet, you can see effectively this basically rolling. Cells on the top go faster than cells on the bottom. So in the reference frame of the droplet, the cells move backwards, and these are the cells that are eventually left behind. And now,
So where is the growth?
I'll tell you now. Okay, I'll share another document. Unfortunately,
Doesn't vary in the red direction.
no, we assume because it's very thin, we assume this kind of flows freely. Okay, I'll show you. Unfortunately, I didn't have them. Well, I have another presentation, but so we have the bacterium, which diffuses with very little diffusion, to be honest, and then it's consumed. So H is the height of the droplet. So the bacterium are consumed at the rate that depends on the height of the droplet. Then we have the chemoattractant that diffuses. This diffuses with as a larger diffusion coefficient. It decays at a certain length, and it's produced by the bacterium at a certain rate. So these are the two diffusible species. And then this is the equation I showed you before. And then this is a form of S. And the key is that H, so the height is driven by these conservation laws. So we have a flux Q that depends on U, and then we have this growth term that depends, where the growth rate depends on the bacterium. And so this is a kind of assuming this goes on the slow time scale, is actually not playing a role in the traveling wave solution analysis. And then, of course, this is defined on a moving domain, so it's a little bit tricky. But then if we look at Q, which is the flux, is basically just the integral of the flow field of the cells. This is kind of standard conservation law, and we get to the standard lubrication thin droplet kind of equation. So it's basically all boils down to conservation law plus diffusion of all and production of all of the other species. No, we don't think is because you're modeling it as a fluid, so you kind of expect that on average is not kind of spreading apart. Diffusion would make it spread apart. So on average, you assume that that's not the case. Because you have surface tension that keeps it together.
No, there's some noise on the hat, no?
Yeah, like it's not going to be perfect, but we average a little bit. Let's say. I think like the key idea is that surface tension is keeping the cells together.
And we see the red some data. And then you have also a data. You don't know.
I think this is like this is more standard in the fluid kind of lubrication limit approach. But I mean,
It's a bit different to what you showed on your slide. Is that part of your expression for you think? Or have you integrated the
it's integrated in H.
So
It's a
from
conservation.
went away. I mean, some of the higher derivatives in your original statement you have gone away.
They are captured here in F. Sorry, F is the driving force. Sorry. So yeah,
What's the F?
F is basically. Not in the simulation before, it was
Okay.
Yeah.
surface tension and the
Yeah,
S.
yeah.
Sorry. Yeah, I know it's I'm sorry. I should have given you more information in the slide, but I don't really- yes, F itself depends on H by the surface tension.
Yeah.
A fourth derivative, yes, because that's third, and then you have Q is derived again.
Oh, yeah, I think just if it was clear that I had derivatives in it, then.
Yeah, no, but I should probably write it better. But yeah, so the key idea is that Q itself depends on the H.
Yeah.
So you do have the fourth order derivative, but you don't have a diffusion second order derivative.
Sine of H by Y.
Yeah.
Differentiate this.
Oh, that with the notivity. No.
Well,
In the,
when you do DQDY, because F has got a DHDY term in it, you only have a diffusive term. But we've also got, so.
Oh, it's a very fourth order.
Can you show it?
Dominant.
Yes.
The linking order is fourth order.
Yes.
But there will also be a second order, a diffusive term.
On the side.
Because you've
On
got
side.
the side.
Psi this is van der Waals.
Yes, so size.
Okay, yeah.
H. So when you differentiate, it, you will get a DH by DY, so there will be.
Yes, there is some effect diffusion. Yeah, but it's actually negative diffusion, so it needs.
Yeah.
No, it isn't, but size
It-
increasing.
Yes,
We get a minus sign.
yes, so it will be,
So it's really-
yeah. Honest,
the head and safety.
Okay.
it's all good.
Yeah, okay. I think like the key is that of that term it actually leads to the instability that leads to the split, basically. It's van der Waals forces. So yeah. Okay, I haven't thought about it in that way. But yeah, so yeah. I guess the key idea is that the gradient is these activities leading to the transition from the compact case in which surface tension and activity basically balance. And then, so what we think is happening is that in the in the first phase of the migration, we have these two terms kind of balance out, but eventually because of you get a lot of mass on the back, it's the information and doesn't move. So the two terms cannot balance out each other anymore, and these terms are okay. And so eventually leads to the split. So we think this is happening. Do I love this model? No. I'll tell you. I think it's nice, it leads to it. It's very nice from a physical point of view, but I still like don't fully I mean, this is a way of modeling it and it works and it's nice, nice representation diagram and everything, but really like what these cells are actually doing and how they're interacting. That's where like there are lots of questions.
This one give you the periodic arrangement of
well, we can basically this is fine because I can have multiple droplets coming in together, so we can capture the periodicity, and so the periodicity is determined by the growth of the cells, because you slowly increase, then you reach the critical mass, then you go back, then it starts increasing again. So we can get the periodicity, and we can get the basically distance between shedding by playing around with the parameters that are how we can feed. But I think like the key missing ingredients, and there's a PhD student that is working on ADM model, on an agent-based model, is like where it's actually is activity. What we think is the interaction at the micro scale is really described by this form of the activity, and really how these like the individual cells are actually moving and like.
Do you think that the scenic characteristics of the cells is changing as they become more style?
We don't think so. So this is the key. The key is that we don't think there is any phenotypic changes, and that's what's also interesting about this system is that you don't need chemistry like biology on top of like physics, let's say. Well, you have some biology, but we don't think that phenotypic changes or gene regulation or something is playing a role in differentiating between the back and the front of the droplet. These are all cells look the same, they're just moving around and eventually some are left behind just because they are there, just because they are on the back.
What was the typical time scale for the movie you showed in the beginning?
So they were the cells?
Yeah.
The experiment I think is an hour. It's over an hour. So they're moving into the
This is why I say they vary how much bacteria they give and the different sizes of the drops.
So
Because you can predict that, I guess. You can fit your model, tune it to what they see, and you can predict what will happen.
yeah, that's true. So the idea is that in terms of the bacteria, I think they try like that is not always perfect, so you not always the same. But I think most of the experiments are analogous. So we haven't like tried different concentrations and everything. Also because we kind of the key idea is also like what are actually these parameters? We don't really like what is the surface tension of the system. Well, without having like a individual cells model, like a clear understanding what's happening at the cell scale and how does that translate to the macroscopic scale, we kind of fit it through the experiments, and so we do like all of the other parameters. And I think for now what they want to do is just like we see all of these like really good stuff. The model kind of reproduces it, predicts it. What we did is that initially, so we did the model, we got the flow field, and then we checked also the flow field. So that's kind of the step in which we do kind of a prediction in some way. But yeah, like down the line, it would be interesting to see other settings. And we do see a different, when you look at the trail compared to the load, you do see different way of shedding and also the load. So in one case, when you have the load, basically the shedding is, when you look at the side view and you look at the shedding is something like this, and then eventually it breaks. So it's much more compact.
Well, it's not moving.
Yeah, it's
I mean,
this way.
it's not like the sharpening static front of the separated things, they are actually moving.
Yeah, this is all moving at the back. So it initially is moving like a. So you have initially that is a compact droplet, then eventually kind of does something like this, and then eventually.
So it's all split into
Two. Yeah, and then
the
it's.
back one stops moving and.
Yes, exactly. So compared, so the process of this, the time scale of this.
The back one stops moving because it has no food.
Yeah, exactly. They have no,
Yes.
they have no information. And then instead, when you look at the single loan, like at the single kind of strip of bacteria, what you see is that they're moved, like it's not like elongated, and then eventually you get the back, and then it's something elongated. So they kind of, they're left behind from the back more than having like a sharp transition. And the model can capture both behavior.
So what do you think that you will make?
We can see. I guess if you ask me like to write down a model for this system, this is not the first thing I will write down.
There are a few things that I would be very intrigued
Yeah,
by trying to make your model fit. One is the velocity gradient that's measured.
yes, we do it.
You have the 12 micron per minute. That's very precise.
Yeah.
And then the whole gradient of the velocity field.
Yes,
There's another one is the length scale.
of the, yeah, and yes,
And
exactly.
with that two pieces of information, you can have a lot of your parameters.
Yes, I think the only one we need is the ratio between the surface tension and the active turn. So the two kind of effective parameters, we can identify them independently. We can identify the ratio, which is
Saying a different angle did something if you were to have.
so I tried
an
to have
experiment
to infer with S.
to infer what, to break the sort of K over Z or whatever.
And not this kind of sort of experiment. Maybe you can talk the droplet in some way and make it change shape and then see how we relax back. But from just looking at the shedding, not really.
Maybe you could put, sort of, drop some bacteria and do something,
Yeah, there's something we can.
some sort of intervention, sort of see if you could, break. Break, And what one would you write down? Don't like this one.
That's another good question. What we did, we started actually with the phase field model. So we started a little bit more
Why
complex.
did you not like continuing with the phase field?
Oh, the phase field, it's really nice in simulations, but you can't do any analysis.
Right, okay.
So it was like, I think the issue is that the phase field model had lots of parameters. And so when we started saying, can we reproduce what we see in the experiments, then you're left with what is actually what the parameter doing. And phase field simulation, like in this scale, don't take like seconds. So I think like we moved kind of to the limitation lead kind of approach to kind of get an handle of what each of the term that was remaining was doing. I guess start simple, I guess. So, but yeah.
So we have done a lot of modeling of cell adhesion, so how cells adhere,
Yeah.
so you get people that move around and so on. And so maybe it would be nice to use the adhesion model kind of device.
Yeah,
The adhesion models are non-local models, and if you expand the non-local terms in moments, you get a first order term and a second order term. So maybe that's the connection I can be made.
yeah, that definitely. And I think, yeah, so I think like there is capillary flow. I think like most of the work done by Camillo and others, like on all of the people in adhesion, that were modeling adhesion between tissues and so on. I think this is really nice. These are not at all. I think in terms of the this active pooling, that's the one that I wouldn't know like how to I'm more perplexed on how to write it down at the emergence. Like maybe at the individual agent-based model, one can be more clear how these things are acting.
Having in mind that if you try something which is just more of a practical thing, it doesn't work. So for example, the procedure of C multiplied by that doesn't work.
So are you in prime colors?
Yeah, these are sort of non-linear, syntactic, some sort of refractory period or, you know.
So I
Have you
think
tried anything else or what made you try this in particular?
So I think in color seagull, the only issue we have is that, I mean, it could be a color seagull class surface tension. But I think that, yeah, but I don't think it would make too much of a difference at the end because you would have still quantified color seagull to separation because these cells cannot, like the velocity of these cells is not just proportional to the gradient, because eventually there is a maximum velocity they can reach.
Well, I also wonder then, but if you look at their respective kinetics and if they're fully saturated and so on and so forth.
Yeah.
But I don't know, I just don't understand the physics behind what you're writing down there.
Yeah, I think the key idea is that if you have to, what this is doing is like, there are models of myelin actually that work like that. So the idea is that you have a cell, and you have another, sort of like what is actually first. There is a cell and another cells, these cells form an adhesion. These cells want to move this way, so it pulls on the cell. So there is like a dipole. And by doing so, it can move itself forward, but it moves this one backwards, basically. Yeah. So there is this dipole, and the activity is basically a matrix of like how strong this active force is. So that's all the current structures. And then it says, but the force at which like the direction of this adhesion depends on how strong. So if I have a clear gradient, I want to move this way, but if I don't have a clear gradient, some cells are going to try to move this way, some cells are going to try to climb on top of the other, and this one will try to learn this way just because they have no idea what they're going.
I still don't understand why that gives you whatever you've got d minus y squared over 1 plus d minus y squared over whatever.
This one?
No, the
This one?
Yeah.
So basically, effectively, what you have is that you say there are dipoles, and the fact that there is this dipole creates a stress, and the stress has a standard form, which is psi, the activity, the strength of how aligned they are with respect to your direction field, and then there is the tensor product between the this polar vector. So basically it says like what's the direction of the line, and here we assume that basically since there is like maybe the direction of inverse is like the direction where the bacteria are y, so please y. Okay. And then you just have like basically newtons,
so some calculation that's under the bonnet that gives you that.
Oh yeah, sorry. So this is basically coming from conservation of momentum. So you have a fluid term where this new comes from, the viscous term, and then you have the capillary for like the capillary, basically another stress part that comes from this capillary term. And then you have stress, then your balancing goes out. So it's balancing off.
Yes, is the numeric order.
Yes.
Yeah, function for it.
So as I said, we use a kind of separating function. I played around with different separating function, and then by changing the parameter, you kind of get, as long as it's separating in some way, then you can play around with the parameters and make it happen in the right regime. But I guess like, yeah, but I think in principle it should be more like this, from what they say experimentally, it should be more like on this switch function. So these cells, basically what they observe is that if there is no gradient, the cells will start moving randomly because eventually, because of fluctuation in the binding of the cells, eventually there's going to be a break of symmetry that leads the cells to move the either left or right without any clear like alarm directions. So the idea is that that's why it goes to 0 when the gradients are too small, and then as soon as they are at a clear signal, then they just start going for it. So
So
that's
it's very, very standard in chemotherapy modeling to use a hyperbolic tension. And this looks exactly.
yeah, we do it.
But I just want to say that example in the literature, but it's actually done.
Oh, okay, yeah. Okay.
Hyperbolic tension explicitly managed of the way that and then shifted so that it's going to do it. So
Yeah,
there's
this is
there's
more-
literature for that, yeah. There's a paper. Yeah. Fine, so we will already.
Okay, because yeah, I think like the key thing is also that then it depends. So effectively, and it's nearly 100 wood is that effectively the movement of the cells, unlike in standard chemotaxis model, does not depend actually on the gradient, but on the curvatures on the second. Because you take S depends on the derivative. And so when you take the derivative again, you're gonna get S prime. And then the second order derivative. So what this is saying is that actually if the gradient were to be linear, there would be no effective movement of the cell, which is in line with our model, but I'm not sure whether from my like from the work that I see, in taxis is a little bit like less standard. Because usually you would assume that the velocity would depends on the gradient directly and not on the curvature. But I guess because of the conservation, like this idea that the force depends on the gradient, then actually gives you that the velocity depends on the curvature, which might be just because of how these cells are interacting and the fact that they can't swim, for example. So I think that's another kind of part of the model that maybe we could test this, but I'm not sure we can control the system where the perfect is.
Is there any way to manipulate the number of sectors or the whatever that kind of thing?
So what they were actually doing in the predictive experiment they wanted to do was to use a mutant to increase the adhesion, the strength of the adhesion, so that they would basically repress the speed. They did some experiment, but the problem is that they're yet to. So whenever you mutate this thing, lots of things happen, and I think that they had a good understanding of what else it was they were changing about the mutant.
That's something that law for the sector.
Yeah.
I think
It's a thing.
there could be, but I think like at that point, we wouldn't even see them coming together in the first, like I think it would affect a lot of parts of the experiment. So the initial coming together and then movement. Like I don't know what this sounds like, really better.
Yeah, you that is like.
You mean like a negative?
Yeah.
Right. Like when they start.
That could be this way, it's done, right, we just to change, to provide the gradient, a linear gradient versus a hyperbolic gradient, versus a step one.
The problem is that like
Whatever is the signal, it gives that, it gives the gradient.
I think the issue is that there is then the feeding. So you can control that initially, but then the cells dictates. And if you have a standard like proportional consumption, they decide, kind of these cells decide what the gradient should be. So that's why it's
Maybe once it started moving at the other end, so they're all moving this way, we've got lots over there. And you can a little bit, and then sort of try and
see where they go.
Yeah.
Another thing that they're interested in doing, this is completely like other, like it would not be a gradient, I think. Kind of testing the model, but it's like now they can do these stripes and then start making bifurcation of the stripes and so see what happens to the droplet. So it is, as they arrive, they kind of need to make some decision. So would the droplets split or would surface tension keep them together? So this is from the top view. And then start playing around like and make this one bigger, this one smaller, and then stuff like that. But that would be again like another layer to the model. So it will not be. I think what we were hoping for was like the Newton version. But they got some experiment, but they were not good enough to go to. They didn't have control enough to make it publishable, I think. But I did see like that eventually the split was suppressed, and that's a prediction of the model as well, because as I told you, there is the bifurcation, and we see that if we crank up the capillary flow, basically, if you look now at to the bifurcation diagram, when you have the mass and you have the surface tension over the activity. You can follow the position of your bifurcation points in the smooth plane, and what you see is that they kind of branch out, but then eventually they come up again into the cusp point. So we kind of saying that if the surface tension will be strong enough, then we would suppress whatever is happening. So these cells will keep on moving as a compact droplet. But again, I guess for me it's easy to say with the experiment, but it was all when you want to publish them, they're very important. Yeah, I think you can do a lot of things of Dictyostelium cells, but as soon as you change something, like we were saying, CIAMP is another important signaling, so you risk that they kind of change completely the phenotype of the cell, and then they do whatever they want. So yeah, I think that on the one hand, they're really like nice systems working because you can do whatever and then you survive. But at the same time, they can survive everywhere, so they will adapt very easily.
And how well is the open-based model producing all of the results?
We're not there yet. Okay. So it's kind of at the moment, like it's more like forming the droplet and getting like the right contact angles and see like there are lots of choices also in terms of rule of how this system is, like how these cells are interacting with each other.
Individual cell.
So what they're doing is
And is there any prospect of taking this continuum of that and taking the depth of it
I think that would be lovely. Yeah, exactly. I think because in data-based model, yeah, there are lots of things to be you need to be careful about. So the idea is that we do have like cells, and again, here is also like what should be the basically the shape of the cells.
Yeah.
And so you have basically cells that they interact with the surface and they interact with each other. The interaction between each other is via a potential, an attractive potential that gives like the surface tension, and I think they use Lanajon's fluids basically. And then you have also in addition is this active pooling. And so what at the moment we're doing is that just to start off is that we don't yet have the chemical fractal, but we just say there is a bias towards the right. So if I'm here, I choose in my certain neighbor cell, then I attach to it and I push it, and I add active pushing force. So you basically have a balancing of the forces where you have some sort of short range repulsion, long range attraction, and then you have the active force that tells you what you want to move, and then you add some noise in the basically orientation. No, well, if it's subscribed, I'll know. If it's they tried also making that a little bit more elongated to allow them to kind of swim one over the other. And in that case, then you have some kind of randomness in the alignment of the cell. But yeah, there is a lot going on, and even modeling the surface is not that simple, because you need to decide whether they are lying on the surface, how strong is the interaction with the surface compared to the rest. But I mean, it's a first year PhD. He's doing great. He's doing an amazing job. But I guess it's a tricky one. So, but yeah, that would be lovely then to move up from here. And that's why, like, where would we that actually? These are like probably like if we engineer the ADM problem, the ADM system properly, then these are the effective equations that we would get. But anyway, you will get an angle on what would be the surface tension and what would be the actual activity if you know what's happening at the micro scale. But I mean, it's really interesting, and yeah, I was really fascinated by the data that they had. So it was like incredible.
And what they about? What I happy?
They're very happy. They're pretty happy. Yeah, I have to say you is very happy.
What is happy?
Yeah. I think for them is the idea that the system is really nice. The fact that the visual forces are playing a role, and you don't have any phenotypic change in the cell. So this is happening without having to involve like much phenotypic changes and everything. So I think they really like it. They like that we can get this sleeping. Yes.
In the experiments, do they really put a gradient of bacteria as a condition?
No, so basically I think what they do.
I am asking because of what insert of the experiments that the cells can make their own gradient.
Yeah. No, there is no. So basically they put like here something.
Uniform.
The uniform alone, I guess it's basically as uniform as they can control it.
Okay.
I guess. And then here there is like just
And
the
they
top cells.
they did their own gradient.
Yes. So they actually, so they can image. I haven't shown it here, but they have. So they image also out of the bacterium. So actually,
That's why I'm asking.
we can get the actual.
In the model, you have to put an initial theme of capital D.
Of capital B. They get yes. I have to. So you can do this one. Like you can start with a symmetric case, and then you can have some kind of inside function, or you can just like. I played around with it. So basically, you have a part of cells, and then you have like almost every side in this way, and then the reflective symmetric and then side in this way, and then eventually I just focus on one side of my simulation because here you kind of close to 0 plus. But yeah, the initial conditioning itself. So if it's true, is that the initial condition do determine the initial dynamics, but then eventually it's kind of converging in the same cycle. We just need to wait kind of long enough.
I think it's probably to leave the room.
Yeah.
Something probably happened.
Thank you.
Thank you so much.
Thank you.